In this session, we are going to discuss the steps of transcription, the main enzyme of transcription, and the post-transcription events. So let's start with transcription steps. The first step of transcription is called initiation, then we have elongation, then termination, and those are the same steps for translation as well. Where does transcription starts? So to recall that transcription is creating a messenger RNA out of DNA. Okay, a segment of DNA is called a gene, and most of the DNA, almost 98% of the DNA is non-coding. It means 98% of the DNA doesn't code for any information. The only 2% of DNA codes for proteins. And when transcription takes place, it will only take place for those coding genes that carry information for making proteins. Okay, every gene has a promoter region and it has a terminator region. So the promoter, it's the start point of transcription, and the terminator is the end point of the transcription. And between the promoter and the terminator, there is the coding gene, the gene that is going to be transcribed. So this is regarding how it starts and how it ends briefly. Now, who is going to transcribe the DNA and make copy of the DNA? It's an enzyme called RNA polymerase. If we go back to DNA replication, we spoke about DNA polymerase. So it's the same molecule. Polymerase is an enzyme that creates a polymer that adds many units to form a long molecule. The difference between DNA and RNA polymerase is simply the type of nucleotides it is adding. Remember that RNA will have U instead of T, and RNA as well is single-stranded molecule. So what is going to happen is RNA polymerase is going to scan only one strand of the DNA molecule, which we call it the template or the coding strand, and it's going to create a single strand messenger RNA. But instead of T, there will be U, which is uracil. So this is called the elongation step. So the initiation is going to happen at the promoter site. Then elongation will take place by the action of DNA polymerase that is going to scan the DNA with the help of helicase that opens the two strands. Then DNA polymerase is going to put the bases in the sequence, correct sequence, which is complementary to the DNA. Then after that, we have at the end region of the gene a small fragment or sequence called the terminator. And the terminator marks the end of transcription. So in brief, initiation starts in a promoter region, then elongation happens by the action of DNA polymerase. After that, termination will take place in the terminator region. And this is the 3 steps of transcription. Now, what is the end product of transcription? After transcription ends, we will have a short segment of RNA. We call it the messenger RNA or the transcript. And it's going to carry the same information as in the DNA, but in a single strand. To speak briefly about the post-transcription events, what does this mean? It means after the messenger RNA is created in the nucleus, it is not ready yet to leave the nucleus. There are 3 steps that must take place in the nucleus before the messenger RNA will leave. What are those 3 steps? If we look at this messenger RNA here, this is a newly formed messenger RNA. It will undergo processing, or we call them post-transcription event. Post, it means after. So the post-transcription event is going to create a mature messenger RNA. What are those events? First, if you look at this messenger RNA, there are dark regions and light regions. The dark regions here mainly are called introns, and the light regions are called exons. So introns and exons. What does introns means? Introns means intruders, as if they are intruders. So those are segments in the messenger RNA that don't code for any information. So they are non-coding fragments or segments. And they have to be removed so that the whole segment of messenger RNA is meaningful and codes for information. So this is the first processing event. The second one is additional of poly A tail, additional of some bases at one side, and something called capping at the other side. Now, more information about processing you will take in the AP course later. So just you have to know that there is a step called processing, where messenger RNA will undergo some changes in order to become mature. After that, this messenger RNA is ready to leave the nucleus into the cytoplasm, where it is installed in the ribosome in order to start translation. So, after messenger RNA is produced and processed, again, what do I mean by processed? I mean the 3 steps of modification. In the nucleus, it leaves the nucleus to the cytoplasm. In the cytoplasm, the messenger RNA is installed in the ribosome and translation starts. So this is the ribosome, this is the messenger RNA. So after transcription is done, now we are going to move into translation in the next session.